What if you could actually detect stress molecules like cortisol using a glowing biosensor on your skin, or use bacteria or fungi to grow living artifacts made out of biological cements? What if you could engineer proteins from tardigrades and use those as a gene therapy to help protect against ovarian cancer, or help to make lab-grown meat more affordable and more sustainable? What if you could engineer probiotics to help confer disease resilience or temperature resilience to coral reef ecosystems? These may sound like science fiction ideas, but these are actually student final projects for the course I teach at MIT. It's an MIT Harvard Global Synthetic Biology course called How to Grow Almost Anything. In How to Grow Almost Anything, every single week we teach you skills and concepts at the forefront of synthetic biology, which is a realm of biological engineering where we're revolutionizing the ways that we can make the medicines, the materials, the food, the energy, and the sensors of the future. And so it's been a real honor to teach this course with Professor George Church from Harvard, Joe Jacobson from the Media Lab, and many faculty from all around the world that are leading experts in their fields. We teach you, again, skills and concepts every week in this experiential lab course. And we teach you how to read, write, and edit DNA, how to design protein molecules, starting with the fundamentals all the way to using the latest artificial intelligence tools like AlphaFold, RF diffusion. We teach you about how to design and build genetic circuits, which are large pieces of DNA. When you put them inside cells, the cells can actually behave like circuits from electronics or computer science. We learn all about cell-free systems, which can help us understand the origins of life on Earth, or they can be used in applied ways and freeze-dried and used as sensors to detect pathogens or pollutants in fabrics and textiles. We explore the frontiers of genome engineering, so how to build completely synthetic genomes from bacteria or yeast. And one of the modules I'm most excited about is around lab automation and cloud biology, which I think is a huge part of the future of synthetic biology learning. And I'm going to play a video that was made by Ronan and Michelle and Suvin, who are here today.
How to Grow Almost Anything is an MIT, Harvard, and Global Synthetic Biology class.
Each week, students learn a key skill in bioengineering, ranging from topics like protein design to cell-free systems, bioproduction, and CRISPR.
This week, students learned about lab automation with the Opentron's liquid handling robots.
Students programmed the robot to dispense fluorescent E. coli bacteria into the shape of a design that they created.
After growing on black agar plates for 24 hours, the living artwork glowed under UV light. Here are some of the amazing designs that the students created.
If you're interested in learning more about synthetic biology, check out HTGAA.org.
What's even more amazing, so the students can program robots to design art, but also even to build genetic circuits and other projects I'll share later. And what's even more incredible is that you actually don't have to have a biology background to take How to Grow Almost Anything. So we have students that are computer scientists, mechanical engineers, urban planners, management students, artists, designers, almost any type of creative or technical background, you can be a student in the course. And the course has really grown tremendously in recent years, more than doubling our number of accepted students to almost 1,300 from MIT, Harvard, and all around the world. And some of our students are even high school students or even retirees that join us each and every year. And so the course wouldn't be possible without this epic group of TAs. A number of them are here in the audience today. It's so great to have you. And the TAs, the vast majority of them, almost 100 in the past couple of years, are actually former students. So you join the course as a student, you become a TA, and some of them even become faculty members. And so it ends up becoming this really virtuous learning community where all of us can share ideas, be inspired by each other, and this grows only more epic each year over time. And the students, at the end of the semester, they share their work in a student final project, which is something personally meaningful to them that represents their abilities and skills in growing almost anything. This is a project from Pat, who made a wearable microfluidic optogenetic device for producing molecules on demand for space exploration. And this is Pat prototyping that device in 0G. One of our former students, Yishao, had never been in a biology lab before and ended up becoming the CEO of her own biotech company, making desktop biology labs. Eyal's project in DNA canvases is a big part of his doctoral thesis, which he's wrapping up now. Belen was a management scientist at MIT and made a whole cell COVID-19 biosensor when I was developing that for her project during COVID. We have had so many architecture students over the years figuring out ways to incorporate living organisms into architectural scale artifacts and objects. Sophia from this year's class made a biosensor incorporated into kite design materials. So again, so many amazing projects. Again, sensors for detecting stress molecules, low-cost modular lab equipment, proteins that can be computationally designed to break down plastics, or textiles incorporating organisms that are sensitive to temperature and pressure, spider silk spacesuits. I mean, really, really remarkable work. Proteins for sequestering carbon. Again, go check out the website. It's a huge set of projects that to me is so inspiring and gives me so much hope for the future. This project from Andreas, who's a city scientist designing sensors that are part of city networks. And this particular project was around making a bioreactor that could grow live cell biosensors. And so he open sourced this bioreactor. It was a big part of his master's thesis, and we're publishing a paper on this. And then this project lived on. The next year, Corinne, one of our students from Hawaii, she used that bioreactor to actually study microorganisms that are native to her indigenous Hawaii. when we were in the pandemic, we were faced with this big challenge, right? We've got this wonderful hands-on experiential lab course, but the students couldn't be in a wet lab. So we did what any good MIT nerd would do. We taught our students how to program robots. And so they all learned Python, they programmed robots, and it could be anywhere in the world. They could be across the street in their dorm room or in Accra in Ghana, London in the UK, wherever our students happen to be. And this is Eyal with one of our robots here at MIT. And we started to realize that you could take classes and lab modules like biological production, where organisms are being engineered to produce molecules like lycopene and beta-carotene, and they could be programmed totally remotely. And so this got us thinking about the future of learning in synthetic biology. How could you actually unleash creativity all around the world, expose students to these transformative learning experiences with only an internet connection and a computer? And it's really mediated through robotics. So we started not thinking just about how to grow almost anything, but how to grow almost anything almost anywhere. And so we took our whole curriculum, figured out what parts could be done with robotics, what parts could be TA-assisted, which ones could be required to be in person. And we started thinking again deeply about how to unleash this creativity all around the world. To me, there's a kind of core story that's really important to me, that this idea that everyone is a creator. All of us have creative capacity to explore and get excited about biology. For us, one of the big things we did later was to start to articulate all this. And we published a paper a couple of years ago now articulating what this global synthetic biology education model could look like. And one story I love, the majority of the authors of this paper were actually students. And a number of those students actually had very, very little biology background. So imagine, not knowing the difference between a DNA or a protein, and then a year or so later, you're a co-author on a Nature Biotechnology papers. And so for us, this big question is about scale. How do we get to, say, 100 million learners all around the world engaging and exploring creativity in synthetic biology? And to me, this idea of radical access is so important. How can you only do it with, for example, just an internet connection and a computer? And again, robotics is such a huge part of that. But I think fundamentally, there's two core ideas that need to be brought together to realize that vision. One is this idea of scalable learning networks, right? Nodes of sites all around the world where students can share, learn, be inspired, build authentic relationships with each other, and then also leveraging robotics, cloud labs to help power and drive that accessibility so you can only need an internet connection and a computer to engage in transformative learning. And so I mentioned this growth in students, but we also have a network of these physical nodes all around the world. This past year, we had sites in New York and in San Francisco, in Quito, Ecuador, in Victoria, Canada, Ottawa, Canada, in London, also in South Korea, and in Tokyo, Japan. And so if you happen to be close enough in person, you could take the course in person, or if you happen to be in the same time zone, you can learn with a virtual cohort at the same site. So you get the benefit of the peer interaction and all the authentic relationship building that happens there. And so for us, one of the real exciting visions for the future is thinking about global collaborative research. How do we leverage the entire network to collaborate and do innovative science together? And so last year, we started a project on global antibiotic resistant bacteria. So one of the big ways to address this big emerging health threat is using something called the bacteriophage. And so you can design what are called lysis proteins. And so we had students all around the world designing their own lysis proteins, which we were then testing locally at MIT using a technology called digital microfluidics from one of our collaborators, Nuclera. And then what we're working on now is enabling our nodes all around the world to prototype and test these proteins locally using low-cost liquid handling robots. This is an example of digital microfluidics working. Each one of these pixels that you see is a different DNA design that's being expressed using cell-free protein synthesis. And every single one of these pixels then leads to a protein design which can be tested. And so this is data showing that many of the students' designs express better than the wild type, which is very exciting. And now we're again working on leveraging this network of robotics to enable the decentralized manufacture and testing of these protein designs. And so this whole architecture points to a system where you're going to have a supercore site at a place like MIT that's got infrastructure, it's got flow cytometry, got mass spec from collaborators like Waters Corporation, flow cytometry, microscopy, and also, again, this decentralized network of low-cost robots. And you can share protocols throughout the network and also experimental data so that all of us can collectively be working on science together. And you can imagine experiments where we could do large parameter exploration, all sites exploring many, many different designs, or do reproducibility studies where the whole network is trying to run the same types of experiments. And so this year, we're launching a global project on what are called neuromorphic circuits. So these are genetic circuits that when you put them inside of a cell, the cell actually behaves a little like a neural network from computer science. And so the students, again, design their own neuromorphic circuits, program robotics to actually build those circuits and transfect those into mammalian cells. And then TAs then studied and got the characterization data. And so this is, again, a project that students all around the world are now starting to contribute to. And for us, biosafety and biosecurity is really important. We were excited to work with Secure DNA on synthesis screening, and then also with Professor Kevin Esvelt on ways to study our robotic protocols for safety and security. And as we think about the future, there's this emerging and growing biological economy, both nationally and all around the world, and it needs a workforce. And so one of the real exciting ideas to me is this idea of biological literacy. We take for granted digital literacy and computer literacy, but I think just as exciting and as important for the future in the years to come is our ability to be literate and creative with biology. And so we're really excited to grow out our national network here with some more nodes that are going to be coming on board in the coming months. And I'll close with a little anecdote. This is a picture with myself and Tom Knight and Jason Kelly, who are two historical figures in the history of synthetic biology. And behind us are these 3 robots. And so each one of these robots, you can put any kind of arbitrary biological tool inside. And if you string them all together, you basically can automate any kind of biological workflow. And so this is a cloud lab for biology. And so this is called Nebula. And we're really, really excited to start collaborating with Ginkgo to think about how we can leverage Nebula for creativity and cloud-based learning in SynBio. And so there was a really famous paper in 1971 that was published by my colleague and the former founding professor from the Media Lab, Seymour Papert, called 20 Things to Do with a Computer. So this is back before we understood that we could use computer for things like learning or creativity. And we're really excited to work on a new project called 20 Things to Do with a Cloud Lab to really push the limits of innovation and creativity for what you can use a biological system and infrastructure like a cloud lab for. And so this is all, to me, the future of synthetic biology learning. How we can take transformative learning experiences like category almost anything, scale it through a learning network, power it with robotics and cloud labs. And all of this is for a larger goal of planetary flourishing. We're entering a phase now of climate crisis, biodiversity loss, ecological crisis, and our ability to engineer and work with the living world is more important than ever. And I really hope that we can use platforms like How to Grow Almost Anything to unleash creativity at scale so we can address problems and really meet this moment and help create a more flourishing planet, not just for humanity, but also for all life forms on Earth. So thank you all so very much.